Chapter 2. Classification of crude drugs. Crude drugs are derived from different natural sources like plants, animal, minerals, and microorganisms. For their study, it is necessary to arrange them in a system study. Each system has its own merits and demerits, but for the study of drugs are classified into different types. 1. Alphabetical classification A. Taxonomical classification T. 3. Morphological classification M. 4. Pharmacological classification PR. 5. Chemical classification C. V. Chemotaxonomical classification CT. Alphabetical classification A. The crude drugs are arranged according to alphabetical order by their English or Latin names. Some of the pharmacopoeia and reference books which classify the drug according to this system is A. Indian Pharmacopoeia, IP, England, B. British Pharmacopoeia, BP, C. United States. For example, acacia, benzoin, cinnamon, digitalis. Advantages, merit of alphabetical classification, LOB. It is simple method. In this system, location tracking of drugs are very easy. No confusion as entries of drugs are not repetitively. It is easily and quick to use. If we know the name of drug, we can study it properly. Disadvantages, demerit of alphabetical classification. Honey. This system doesn't help in distinguishing the drug from plants, animals, and mineral source. This system does not show that which entries are new and which are old. The natural of drug is not known whether it is organized, cellular, drug or unorganized, acellular drug. Morphological classification. In this type of classical, the crude drugs are arranged morphologically or externally characters of plant part which are used as a drug, like root, stem, leaves, flower, fruit, etc. They are classed as unorganized drugs. It includes those drugs which are obtained from product of plants, like dried latex, gum, wax, etc. Drugs, animals, mineral, clove, volatile oil, aloe, dried juice. Advantages, merit, more convenient for practical study. It is more helpful in study of drug whether it is organized or unorganized drug. It help to detect the adulteration. It give idea about the source of drug. Disadvantages, demerit. It does not give idea about the chemical constituent and uses. It does not classify whether the drug is made by animal sources or of mineral sources. During collection, drying, and packing, morphology of drug change. Chemical classification. Crude drugs are divided into different groups according to chemical nature of their active constituents. The chemical classification of drugs depending upon TH. Grouped in one place. Type of chemical, example. I. Alkaloids. T. Vinca, cinchona. 2. Glycosides. Aloe, senna. 3. Volatile oil. Peppermint, clove. 4. Lipid. Mustard, peanut oil. 5. Carbohydrate and agar. Honey, gum-derived product. 6. Proteins and enzyme. Papain, trypsin, yeast. 7. Vitamins. Shark, liver, oil. Vitamins, water, fat, soluble. Soluble, B, C, A, D, E. Advantages, merit. If we know the chemical constituents, it is very easy to study about the drugs. Grouped in one place. Type of chemical, example. It is often mistaken for chemical composition in pole manufacturing. Ecological actions drugs. Cardiotonic digitales, squills, Anthelminthix male fern, quassia, antisensor podophyllum, vinca expectorant vasaca, licorice. Advantages, merits. If we know the pharmacological action of drug, it is very easy to study the drug. With the help of pharmacological classification, medicinal importance of the drug is known easily. Even if chemical constituent is not known, the drug may be classificationed according to their pharmacological action. Disadvantages, demerit. If some drugs have two different pharmacological action, then it is very difficult to classify them properly. It does not give an idea about drugs, whether it is organized or unorganized drug. Morphological organoleptic classification. Taxonomical classification. It is a kind of botanical classification and is only. Advantages, merits. Drugs are easy to locate according to their family. It is more scientific method. This system helps in studying of evolution and development of drug. Disadvantages, demerits. This system fails to distinguish between organized and unorganized drug. It does not give any idea of chemical constituents, restricted mainly to crude drugs, which are obtained from plant source only. The chemotaxonomical classification. In this system, the drugs are classified based on both chemical nature as well as taxonomical classification of drugs. Example, plants belonging to Umbelliferae family contain volatile oil. Plants belonging to Solanaceae and Apocynaceae family contains alkaloids. Applying chemistry to taxonomy is known chemotaxonomy. Chemotaxonomical knowledge supports better classification of drugs. Advantage, merits. This classification is R.